All right. Our next speaker is Clemens Messerschmidt, and he's going to be talking about managing high throughput sequencing and other omics data with rodeo and rodeo-ingest. Take it away.
Yes, thanks a lot. And yeah, for the introduction. So yeah, let's get right into it. So maybe the first question that I would like to answer for those that are maybe puzzled what omics data are. So omics is an umbrella term, and it describes a number of branches in molecular biology or biology. So there's genomics and transcriptomics and proteomics. And so, for instance, if you're studying the structure, the function, or the evolution of the genome, then what you're doing is genomics research. And the people who are doing research in genomics or transcriptomics, one of the methods that they use is high throughput sequencing, where they sequence DNA or RNA and then do their research on that. And one of the leading manufacturers of sequencing machines is Illumina, and they come in different sizes and different configurations. And I just put some numbers on the right. So the running times based on how much data you actually generate and how you set up the machine range from like 4 hours to maybe 60. And the output is like millions or billions of so-called reads. So you read the DNA, and those are the reads. And if you then look at the files in the end, like it can go from a few gigabytes to maybe hundreds of terabytes. And that's the size of your output data. And maybe one thing, if you're not really familiar with it, so if you're on the really big machines, then you also need to put a lot of material on it. So you're not sequencing just one sample for one client, but you're putting a lot of samples on it. And what that is called is multiplexing. So at the beginning of each stretch of DNA, you put this little snippet of DNA, which is called an index. And then later on, you can identify which read came from which sample. And this process of sorting out the sequences again then by sample is called demultiplexing. And if you are doing or managing a sequencing facility, then you kind of need to manage both aspects of that. So you need to have something in place to manage the raw data, which is sometimes also called a flow cell or BCL for base calls. And then you also need to manage your FASTQ files, which are then probably the files that you will deliver to your clients. And if we look at the life cycle of data, most textbooks show it like this. So somebody, maybe you, does the data generation, then there might be some data pre-processing, then follows the data analysis, and possibly you're doing some archiving. And in the end, sometimes you also need to get rid of the data because storage is not infinite. And with omics data, it's usually like this, that the people who really generate the data are not the people who analyze it later on. And this is really one of the problems that we want to solve. So usually in these biomedical research institutes, for instance, you have these core facilities or service units or whatever you want to call them, where you have really specialized technicians and with these huge machines or an assortment of machines, which will do the sequencing for you. And then they have the data, and it needs to get onto your computer, be it your workstation or compute cluster, whatever. And the question is how, because it doesn't scale that the clients walk over to your lab and just pull off the data from the sequencer onto a hard drive and then they walk back. Like that's not going to cut it with the data that you really need to move. So we thought about an iRODS-based solution, because it unlocks all these powerful tools for data management. So we wanted to use these automated ingest of all the newly generated data, and then also extract metadata that we get per experiment and annotate the collections. But what's also nice is that we get secure collaboration, which is important if you're dealing with human genomic data. And also what's important for us was that we can get cross-institutional authentication. And yeah, maybe later on, also we want to use the rule engine for archiving and deletion. So you get access to this whole ecosystem, which helps you solve some of the problems. So here's a really simple sketch of how we envision Rodeos, which stands for Raw Omics Data Access and Organization System. So you have the sequencers in the sequencing facility, and they write out their data to some kind of landing zone. For instance, on a network attached storage, or yeah, something like that. And then we do the ingest into our iRODS-based storage. And then from there, the clients can pull their data and only their data. So yeah, what we configured, which data belongs to them, they can then download. So the minimal setup looks something like that. You need an iRODS cluster, you need a landing zone, and you need a host that's running the ingest service. And yeah, of course, that needs read access to the landing zone and then write access to iRODS. And yeah, once you get to like in some settings, of course, like the machines are not all in the same building or even in the same room. So yeah, you also need to think a bit about network, because this is really a distributed system in the end. And I set up a little Docker compose if people want to play around and have a first look. So iRODS-ingest in general works like this. So you ingest the data from someplace into iRODS. Yeah, okay, but what does it do? So usually you extract some file metadata. So if you have some file attributes on some POSIX file system, you can extract those. But you can also parse specified files and file formats for additional metadata, which I will show in a minute. And you can also compute some checksums for each file, right? And yeah, so this is implemented in irods-capabilities-automated-ingest, and we extended it with rodeos-ingest, which provides two, I think the terms event handlers, two Python modules basically, and the one handles these raw files and these PCLs, and where we parse the Illumina metadata and annotate the collections afterwards. And the other ingests directories of FASTQ files after demultiplexing. And what we haven't implemented here is like parsing QC reports or sequencing statistics, which might be interesting, but we haven't really looked into that yet. And the GitHub link below points you to that. And if you look into which kind of data Illumina sequencers generate for each experiment, so usually there are files generated that are called runinfo.xml and runparameters.xml, and they look like this. So it's like hierarchical structured information on the experiment. So you get lots of stuff like the serial number of the sequencer, you get expiration dates for the chemistry that is used. You get lots of information about the whole run. You get software versions because there's also software involved. And we parse all that. And then we put it into our iRODS collection where we pulled in the data. So here I show some of the parsed attributes with iMeta for one flow cell that we ingested. So you can see the date of the run, you can see the flow cell, you can see the unique ID of the instrument and so on. And what we really envision later as interfaces for Rodeos. So we are currently using or experimenting with meta links, but as we heard today, we probably also need to move to the new tool called the zone management tool. And that's mostly for technical staff. And using that, they have an easy way with the graphical interface to manage the client access. So they know which client owns which data, and then they can, make iRODS collections accessible that contain the data. The I commands, of course, so probably people will use irsync to download the data that belongs to them to their near compute storage. So as I said, their workstation or their compute cluster. And then we also want to support WebDAV as an accessible overview, for just about anyone who wants to have a look. And in metalinks, you can see the same metadata that we extracted as I showed before on the command line. And this is the graphical user interface for making the data accessible. So we can have defined groups for longer running projects, or you can also make data accessible for individual users if you want that. And the pilot implementation that we are currently working on, it's a bit more complex than the picture I showed in the beginning, right? So the Berlin Institute of Health is a virtual institute, more or less. So they're actually researchers from two other research institutes. So there's the MDC, the Max Delbrück Center, and they are doing mostly more basic research. And then there's Charité, which is the university hospital in Berlin. And so, every researcher at BIH has an account, like an Active Directory account for either MDC or Charité, or maybe both. And additionally, also the sequencers are not all in one place. So some are physically located at MDC and also in the MDC network, and some at Charité. And we expose a landing zone based on Ceph through Samba gateways for one Samba gateway for each sequencer. And we expose one landing zone, and the central demultiplexing then can run from there onto another landing zone, also based on CephFS, and then we ingest the base calls as well as the fast queues into our iRODS storage. Then somebody needs to give out read access, and then what we use is PAM to integrate with the Active Directory of both institutes. So users use their normal user account and normal password that they also use for email and whatnot to access the data, and then they can download their data. And open questions, future work. We also want to branch out a bit into mass spectrometry data later on, which would be needed for metabolomics and proteomics experiments, which is another core facility who are doing just that. But one of the challenges is that the metadata formats are not as well defined, and there are more competing vendors. So it's a bit harder to get everyone on one page. We're currently not tracking any sample information for these multiplexed runs. I mean, you could do it, but they're probably more accessible tools. Like most sequencing facilities have a LIMS in place already. And we also have a solution to do that with the graphical user interface. And archival and deletion, we haven't really touched yet, but mostly because the data life cycle needs to be properly defined for these files, and there might be even multiple different ones depending on who owns the data and maybe who paid for it and so on. So that's all work in progress. So have a look, try it out, let us know what you think, and thanks for your attention.
All right, thanks, Clemens. Are there any questions for that? Okay, Justin.
Yeah, it's not a question, but if you're on Slack, Mike Conway at NIEHS has, he said he's doing a lot of the same things. Are you on Slack?
I'm on Slack, I'm on the Slack.
Okay, maybe you can just read the comments. I don't have to repeat it here, but it sounds like he's doing a lot of the same things, so you might want to get in touch with him.
Ah, nice. I didn't see that. I will have a look. Mhm.
Yeah, can I start? I just have a simple question. Are you tied to the Illumina sequencers, or you would have to like adapt to a new workflow if you want to use a different brand of sequencers, or it
So-
wouldn't matter?
Yeah, so like you can do the normal ingest without any additional metadata parsing for basically any collection of files. But the parsing of the metadata right now, we only implemented for Illumina sequencers because that's what, yeah, the major chunk of what people do right now. But yeah, if you would like to have other sequencing machines, then that shouldn't be a problem. If you know how the metadata is structured, then you could implement it and add that. Yeah, sure.
Okay, thank you.
All right, John.
Oh, hi there. Just so first of all, struck how similar your approach is to the one we have at Sanger. The very similar layout, probably because we use exactly the same instruments, right?
Yeah.
As well as PacBio and Illumina, not Illumina, Nanopore. But it was interesting. I see you're using CephFS as a landing stage. How are you finding that? And as a follow-on with that, why not use the Librados plugin?
Okay. So yeah, I think that's just because we were using CephFS for some other things, so we stuck to it for this. But so the experience until now is fine. So I can't report any major problems. But yeah, you can probably also use. So for the landing zone, you can use anything that you like, really, that is stable for the sequencers to write to. Yeah, that's true. So, but just for our pilot implementation right now, we are on CephFS. Yeah.
Thank you.
All right.
Hi. I saw that you had an automated workflow to ingest data and metadata from the sequencing machines, and that seems to go through Samba. I was wondering how the contextual information, the information about the context of the experiment, and eventually gets related to the data.
I'm not sure I really got your question. So with this Samba, we are only really using to expose the CephFS as a network share to the Windows-based Illumina machines.
So you'll have some files copy over about some experiment, and the information that can be known automatically, that's automatically associated with the data. But how's the information about what this experiment is called, why somebody's doing it, how does that get categorized and stored?
So, right, okay. So this is maybe more related. Oops, can I go back? So this point tracking information. So I mean, because almost all runs are multiplexed nowadays. So if you just get the raw data, you will not know what was on that flow cell. Like you will not know, was it tumor samples, is somebody doing single cell experiments. So for that, you would really need the sample sheet and maybe some additional, points. I mean, this information is there. It lives in emails maybe between the researcher and the sequencing facility, or we put it into our system, for instance, but it's not actually right now tightly coupled with the iRODS. Yeah.
Okay, thank you.
All right. Thanks, Clemens.
Yeah, thanks for listening.